Mercer and a previous student from the University of Washington's Engage program, and I'm pleased to welcome you to our event tonight. Tonight's guests have the wonderful opportunity to speak to you because of a special collaboration between Town Hall and the UW's Engage program. Together, we host a series called UW Science Now as part of the Seattle Science Lecture Series. Sponsors include Microsoft and KPLU. Now to our first guest. Sharon Greenblum is a graduate student in the Genome Sciences Department of the University of Washington. Originally from the other Washington, as in DC, Sharon got her training in biomedical engineering, studying how human genes interact to cause cancer. Now she's curious how the genes of other creatures affect human health. She uses computers to figure out how the bacteria that live in the human body interact with each other and with us. When she's not studying bugs and poop, she enjoys playing soccer, traveling, and pursuing random cooking adventures. Tonight, she will delight us with stories from your stomach, an interview with your friendly gut bugs. Please give a warm welcome to Sharon.
Thanks so much for that introduction, Juliana. As she said, my name is Sharon Greenblum, and I'm a student in the Department of Genome Sciences at the University of Washington. And tonight, I'd like to tell you a story. Now, the setting for this story is a rather unlikely place. It takes place in your stomach. Because deep within you, there's a whole other world you might have never even known existed. In this world, there's drama, warfare, heartfelt family sagas. Deep within you, there are an unimaginable number of tiny little creatures going about their everyday lives. And though you can't see them and can't feel them, they might be having a huge impact on your everyday life. Let's begin our story. To do that, we have to rewind back to 1683. And here you see Antony von Leeuwenhoek. Now, he looks like a very distinguished scientist, but he wasn't. He never went to college. His main job was hanging curtains. But Antony was curious, especially about the things that he couldn't see. And today, we know von Leeuwenhoek as the father of the microscope because he devoted his life to creating lenses that would let him see the invisible. One day, Antony decided to scrape some of the white stuff off of his teeth and put that in the microscope because why not? And what he saw astounded him. He even wrote a letter about it. He wrote, "I saw a little white matter, as thick as if 'twere batter." He was apparently also a very mediocre poet. In the said matter, there are many very little living animalcules, very prettily a-moving. All the water seemed to be alive. Now, this is a very vivid and possibly overdramatic description, but Antony was really onto something. What were these little animalcules he saw swimming around? Well, today we know them as bacteria. Also, microbes, microorganisms, or sometimes more fondly, bugs. What we're referring to with all of these are tiny one-celled organisms that live pretty much everywhere on Earth. Now, if you think about the big family tree of life, our closest relatives are apes and chimps in the animal kingdom. To get to bacteria, you have to go all the way around to the other side of the tree. They're about as distant from us as you can get. Their side of the tree has way more branches, though. So that means there are thousands and thousands of types of bacteria, and each of them can do slightly different things. You may have heard of some of these types of bacteria, but as with many things in life, it's the ones that get a bad rap that get the most hype. So you've probably heard a lot about E. coli and Salmonella. They show up in rotten, spoiled food. You might have also heard of Yersinia pestis. This is the bacteria that caused the Black Plague. MRSA is a bacteria that affects hospital patients. But there are also plenty of good bacteria. For example, you might have seen the word lactobacillus on the back of your yogurt container. This is a bacteria that's helpful for digestive health. Now, as I said, bacteria live pretty much everywhere. So you'll find tons of them in the oceans, literally tons. And even in the driest, most inhospitable places, there'll be bacteria. Another place you'll find bacteria? You. That's right, every surface of your body, inside and out, is covered in bacteria. And that's where our story gets really interesting. Because what that means is, you are not just you. Each one of you sitting in this room right now is one you plus 100 trillion bacteria. 100 trillion. So if you need to take a minute to kind of go like this, that's fine. But I'll just let you know that these bacteria have been there since the day you were born. And if everything goes well, they'll be there for the rest of your life. And that's actually a really good thing. Let me just take a minute to convince you what a big deal these little guys are. So first of all, as I said, there are a lot of them, 100 trillion just living on you. Now, each one of them is just one cell. But do you know how many human cells you have in your body? Only about 10 trillion. So when you count up the number of cells, they outnumber you, 10 to 1. How do they all fit then? Well, they're tiny, teeny tiny, about 1/1,000,000th of a meter each. That means that about 50,000 of them might be able to fit on the head of a pin. This is an actual picture of the head of a pin, magnified to huge proportions by a really powerful microscope. So you can see individual bacteria, and they're still tiny, and there's a lot of them. That's about a packed Sounders Stadium worth of bacteria that can fit just in that space. Now, as I said, there are a lot of them. So even though they're tiny, you can't ignore them. And when you put them all together, they take up some serious mass. So if you took all the bacteria that live just on you and put them on a scale, they'd weigh more than your brain. That's something to think about. Now, the most amazing thing about all these bacteria is what they can do. So the most important bacteria live in your stomach. And there they act as a kind of mediator between what you put in your body and what you're able to get out of it. So for example, bacteria are responsible for digesting some of the foods you eat, extracting energy from them, and making that energy available to you. Bacteria also might be responsible for whether some of the drugs and medicines that you take work or not. And bacteria can act as regulators of your immune system, your body's defenses. They help your body figure out what's foreign and what isn't, and make sure that your body doesn't go crazy attacking the wrong thing, like itself. Each of us, though, has a slightly different set of bacteria. There are different types in different amounts. And that means that your bacteria can do each of these three things in slightly different ways than other people. So in a lot of ways, when you think about it, our set of bacteria is more personal and more important to us than, say, our appendix. And in that case, it's really worth asking, who are these mysterious creatures we're carrying around with us every single day? How are mine different than yours? What are they doing for me? And possibly more importantly, what can I do for them? These are really good questions. But for a long time, they were really challenging. Here's why. So ideally, to study a specific type of bacteria, you'd want to take it out of its environment, isolate it from all of the other types, take it to a nice controlled lab environment, cool AC, easy living, nothing too difficult, and then subject it to lots of tests to see how it responds. So kind of poke at it in a metaphorical sense. Up the temperature, give it some crazy things to eat, and see how it reacts. Scientists tried to do this with lots of different types of bacteria. But 99% of the time, it failed. The bacteria didn't even make it to the easy living stage. They just died on the spot. What's going on? Why? Well, bacteria might actually be a lot more like us than we give them credit for. See, bacteria form communities, where each type of bacteria is actually depending on a whole assortment of other types for its very survival. Bacteria rely on each other for providing food or signaling when there's danger. Take one type out of its environment, away from all the other types, and it just can't live. This is true of 99% of bacteria that we know about. And I mean, I can relate, right? If you take me out of my community, away from my mom, my sister, my best friend, the place down the street that makes really good pizza, frankly, I just don't know how long I'd last. I really need my pizza. Bacteria are the same. They depend on each other. They need each other. And this makes our life, studying them, really difficult. So for a long time, we knew that bacteria were out there, living on us, living in us, but we had no real way of studying them. And so we didn't know which types lived where and what exactly they were doing. Fast forward though to the 2000s, and suddenly everything changed. Biotechnology was booming, and a new technology came out that let us see exactly which types of bacteria live where without taking them out of their environment. The way this technology worked was by looking at the bacteria's DNA. This is that long string of letters in every single cell of every living thing that's the code or instructions for how to make that thing. Scientists found that there was a certain part of bacteria's DNA that acts as a kind of barcode. So each different type of bacteria has a different sequence of that part. What that meant was that if scientists were able to take a sample of all of the DNA from inside a person's stomach and just look at these little barcode areas, they could get a kind of census, a bacterial census of which types of bacteria and which relative amounts live in that person. This was great news. This really revolutionized the way we were able to study the bacteria that live in the human body. And so over the past decade, scientists have gone into a bit of a frenzy, looking at the bacteria in all different types of people, all different demographic groups. They've looked at the bacteria that live in diabetic people, obese people, people that are elderly, infants, people from different countries, people with different diets. You name it, they've looked at their bacteria. And the possibilities from studies like these were really tantalizing. What scientists hoped was that maybe if they looked at enough people and enough samples, they'd find a specific type of bacteria that could potentially be the answer to the obesity crisis. Or maybe there was a type out there that could cure diseases. Or maybe they'd be able to find a type that would let you drink this beautiful milkshake even if you've been lactose intolerant for years. So, after all these studies, after all these samples, what did they find? Did they find the good guys and the bad guys, the heroes and the villains? Not exactly. This is a story, not a fairy tale. In general, what scientists found was that a lot of times there are more differences between people in the same group than people in different groups. And that made things really confusing. So, take obesity, for example. There have been lots and lots of studies looking at the differences in the bacteria that live in obese people versus lean people. One early study in 2005, came out with this really great result, that when you looked at obese people, they found more of a certain type of bacteria called firmicutes compared to another type called Bacteroidetes. This was promising. Maybe this was where we should start looking for the answer to obesity. But 5 years down the road, another study came out showing exactly the opposite, that Bacteroidetes were increased compared to Firmicutes in obese people. To make matters more complicated, that very next year, another study found that both Firmicutes and Bacteroidetes were increased in obese people when you compared them to lean people. And of course, the icing on the cake, that same year, another study found that there were just no differences in terms of these two types of bacteria when you looked at obese versus lean people. Pretty confusing. There were no clear answers. And that was pretty much the state of things when I joined the graduate department at the University of Washington. Now, to be honest, I didn't know that much about bacteria when I joined the lab. But I joined a lab that studied human-associated bacteria because I was fascinated by the effect that they could have on our lives. Now, I studied engineering, and my advisor was a computer scientist. So between the two of us, there wasn't a whole lot of classical biology training. But we had something else going for us. We were both taught to think of everything like a system. That means not just looking at the individual pieces of something, but actually looking at how they work together to create a whole. For example, when I was in college, I studied all of the muscles that are in your knee and how they work together to help you bend your knee. When we began to think of the bacteria that live in the human body like a system, not just the individual types, but how they work together, things began to make a little more sense. Here's the idea. So to understand a system, you really need two things. You need a parts list and a wiring diagram. Let's go back to pizza for a second. So take this pizza oven. In order to really understand how it works, and maybe even begin to tinker with it so that your pizza comes out hotter or crispier, you need to know both which parts go into making it and how they all fit together. Seems straightforward. Now, when it comes to the system of bacteria that live in the human body, though, we've gotten really good at understanding the parts list, what types of bacteria live in which people. But what we were missing was a way of keeping in mind the wiring diagram, how these bacteria interact with each other, how they need each other, depend on each other, how they form a community. In the end, we realized that we could use this same feature, the feature that bacteria rely on each other and form a community, this feature that had been making studying them so hard for so many years, we could use that to actually better understand them and how they relate to our own health. Essentially, what we needed was a change in perspective. So whereas Van Leeuwenhoek zoomed way in to see his individual dancing animalcules, we wanted to zoom back out. We wanted to look at not just one bacteria, not just one type, but how all of the types fit together. Here's what we did. We created wiring diagrams of all of the bacteria we found in all different people. We then compared these diagrams in two groups of people, lean people and obese people. We looked to see if there are any differences. And what did we find? There were differences. Not in any one specific type, but in the shape of this map of the bacteria. Here's an example of what we were seeing. So in lean people, we were seeing that these wiring diagrams or maps of bacteria look very different than obese people. And if you imagine this diagram to be a kind of map of the bacterial community, essentially what we were seeing is that the maps from lean people had many more neighborhoods. There were sections of this map where there are way more connections within that section or neighborhood than between sections. Looking at the maps from obese people, they look more like what we in the science world tend to refer to as a giant messy hairball. We couldn't find neighborhoods. Everything was just as connected to everything else. This was a really cool result, and we were really excited about it. And then we started thinking about what it might mean. So here's what we are thinking. We're talking about communities here, and you can actually learn a lot about a community by how it's laid out. The maps that we were seeing in the bacteria from lean people look more like a map of Seattle, a city composed of neighborhoods, where each neighborhood is focused on something slightly different. Now, reasoning about why Seattle might look like it does, we can point to the diversity of resources that Seattle has access to. So for example, there are neighborhoods that have a long history of fishing. There are other neighborhoods that are more industrial based. There are neighborhoods that make use of a lot of intellectual resources. There's the tech sector. And then there are neighborhoods that are chock full of cultural resources, places like Town Hall. Moving over to the obese maps, they looked really different. They looked more like something we would see in a town that uses just one specific resource for one specific industry. A factory town, for example. Let's say Steel City, Illinois, a real place. You can imagine that most of what goes on in that town is devoted to taking one specific type of resource and making one specific type of thing. Now, let's go back to bacteria. Where do their resources come from? Well, most of what bacteria have to work with comes from you. The types of foods you eat that make it down into your stomach are what the bacteria set the environment for the bacteria that live inside of you. You're really setting the stage for which types call you a home. And too much of one type of nutrient might cause differences in the types of bacteria that live there, way beyond things like just counting calories. So this is what we're seeing, these differences between the maps of bacteria. And while these are just associations and can't point to a specific way that these bacteria are causing obesity, It's a really good place to start looking at how this system of bacteria is affecting us and how we can continue looking at bacteria in this way and how they're communicating and interacting with each other. We've still got a lot of work to do, but in the meantime, you can ask yourself, are you giving your bacteria Seattle, or are you more of a one industry type of town? As I said, there's still a lot of work. This is just the beginning, but I think we can learn something. What is that? Well, we can change our perspective about ourselves, the way we think about ourselves. We're not just one person, isolated. We're part of a much bigger community. We rely on other people. But we're also a very important part of a much smaller community, one that has just as much an effect on us as we might have on it. So remember to be kind to your communities, and they'll serve you well. Thank you so much for your time. I've really enjoyed talking to you. I'd also like to thank the Engage program for their amazing science communication training, Town Hall for hosting us, and my home for the past couple years at University of Washington in the Borenstein Lab. Thanks so much, and I'll take questions.
I'm curious as to where these bacteria come from in the first place. Are they in a sperm cell and an egg cell, or do they come through the mother? Where do you get the ones inside of you? They must be there when you're born, right?
That's a great question. So actually, what scientists think is that when you're in the womb, you don't have any bacteria. And it's as soon as you come out that whatever you come into contact with first, which is usually the bacteria from your mother, but could be from the nurse who first touches you, those are the ones that get to colonize you first. And that can actually have a lasting signature for the rest of your life. Hi.
So you're talking about the connections between different types of bacteria.
Yeah.
What exactly are those connections? Are they like metabolic pathways that are shared, or?
Yeah, exactly. So the question was, what are these connections between bacteria? And it's exactly what was suggested. We're looking at metabolic pathways, so how one bacteria might produce something that another bacteria uses. Yep.
Yeah, so my question was part of that too. Like, how did you actually figure out those interactions? Because obviously you can't ask them.
Right, right. So what we did, the question was, how are we figuring out these interactions? What we've been doing is looking at the genomes of the bacteria, so which genes they have, which functionalities they have. And we know that one bacteria might be able to convert something to something else, and another bacteria has a gene or a capability of using that and converting it to another thing. So it's an inferred interaction. We're not actually putting the bacteria together in a lab because it's so hard to do because they don't grow very well. But this is an inferred interaction that we believe could be happening.
I had a question following up on that. First, a comment on the, like, are you born with it bacteria? There was just an article in the New York Times today that came out about the placenta.
Yeah.
Did you read it?
I skimmed it, yeah. I saw
and how they found, like, that your placenta as a child, or as an embryo and a fetus is made up of 10% bacteria, and they're trying to figure out where it comes from and all that kind of stuff. And then I think that's important.
Right.
And that was cool. Anyways, I was wondering, how did you study, were you studying like the actual DNA of the bacteria, or do like they also produce messenger RNA? Like, how can you investigate the expression of the DNA of bacteria?
That's a great question. So the question was, are we looking at the DNA? Are we looking at which genes they express, or just which genes they have? For the work that I showed you here, we were looking at the DNA, so which genes they actually have. A whole nother level of this is which genes they express. And so that's other work has been trying to get at that too. And so there have been other studies looking at that type of data. So whereas you might be, one bacteria might have the genes for something but is not expressing it, or might be expressing one gene in much higher levels than another. So that wasn't incorporated into this work, but that's definitely a new step in this field. Thanks.
Yeah, I had a question.
Sure.
I was wondering whether you have any sense of whether bacteria relationships can change over time. You were kind of suggesting that someone who went from lean to obese potentially could be transforming that through their relationship with bacteria. Have you seen something shift over time?
So the question was, are you asking about whether the bacteria themselves change over time or which
I
bacteria
guess
you
the
have changed over
relationships,
time?
When you found a correlation between the relationship or the lines, the wiring, between the relationship and lines for obese,
Right.
do you find that it can actually change in someone if they go from lean to obese, that the wiring will change?
Right. So the question was, does the wiring change of these diagrams over time? And the answer is that since we're looking at the genomes of the bacteria, those don't really change. We have a specific genome associated with a specific type of bacteria. And so that's information we get that's not changing. But what does change is the types of bacteria that could live in a person. And so depending on what types live there, the shape of this wiring diagram might change over time within a person. But the relationships between individual types of bacteria, we would have to actually do that in a lab experiment to see changes. But for the most part, we assume that that's pretty stable.
I have a related question.
Yep.
Which is, in medicine, often someone will be infected, say, with salmonella or E. coli or whatever, and initially they're not sick, and the bacteria replicate, and they get to a sort of a tipping point, and all of a sudden the individual who's infected becomes very, very ill. Do you have any knowledge about how the bacteria communicate to one another that, okay, guys, we've got enough troops, and now we're going to make them sick?
Right. It's
the communication I'm wondering about.
Yeah. So the question was about bacterial communication, and is there a tipping point where a certain amount of bacteria will actually cause the person to be sick? That's not something that I've personally looked at or we've looked at in our work, but there definitely could be an imbalance that's only reached at a certain point. So it makes some sense. I'm not sure exactly how the bacteria would communicate that necessarily, but they do have a lot of pathways devoted to communication. That there are a lot of molecules that they can send between each other that signal the presence or growth of different types. So it's a complicated world, but there are signaling mechanisms between bacteria. Yeah. Hi.
Great talk.
Thank you.
So I was curious about the host end of things.
Okay.
So when you're looking at your system, do you also look at the human genes to see, how that, because bacteria modulate immune responses
Right.
and neurotransmitter release and all kinds of things.
Yeah.
So I'm wondering what,
yeah.
do you see, have you looked at that relationship?
So not in this data set, and that's actually another thing that is just getting started, is trying to compare the genes in the host, the human, to see whether that's affecting the bacteria that live there. And so far those studies have been pretty few and far between. It wasn't included in this work that I was talking about at all, but there definitely could be a relationship between the types of chemicals and molecules that your body is able to produce, and then creating an environment that certain types of bacteria are more able to live in.
Right, because
Yeah.
every individual is unique.
Exactly.
They have different,
Yeah.
and,
So that's another level of interaction between yourself and your bacteria, for sure. Yeah.
Fantastic, thank you.
Thanks.